Hi, I'm Alan Fine. I'm the founder and CEO of Alentic Microscience in Halifax, Nova Scotia. Our company has been developing a new approach to cytometry and other key diagnostic tests to make them more easily, efficiently, and affordably available to people anywhere in the world. What we have set out to do is to replace existing technology that uses complex and expensive machines and highly trained technologists, to replace that with a very simple, easy to use instrument that can be afforded and used by people essentially anywhere. And here's what we've developed. This little device that can fit in a white coat pocket takes the place of benchtop fluorescence flow cytometers, takes the place of benchtop complete blood count machines, of fluorescent plate readers, and several other devices, and in that way can avoid the cost and expense and complexity of those machines and carry out standard lab quality analyses on tiny volumes, as little as 20 microliters of blood or cell suspension from a tissue aspirate, for example, and return results. Well, for blood count, it can give a high quality CBC, complete blood count, in under 7 minutes, and it can do an immunophenotyping, a multi-channel immunophenotyping in under half an hour. And this is done hands-off. The specimen is just loaded into the machine, simple consumable that contains dried reagents and has very long shelf life, is loaded into the device on top of the deposited specimen. The device is closed, and then the operator simply presses go on the machine and walks away. And the machine chugs away and does everything that it has to do autonomously. It creates, after incubating the specimen with the dried reagents, it then creates cellular monolayer, images the monolayer, high-resolution lens-free microscopy, and then analyzes those images by embedded artificial intelligence, evaluates the images to identify the objects, classify, measure, and so forth, extracting the information needed for cytometry, for multiplex immunoassays, serology, blood counts, and so forth. It's all done quickly, as I mentioned, under 7 minutes for CBC, under half an hour for the flow cytometry equivalent, and the device is wireless capable, so it can send the information off to the hospital information system or remote sites anywhere. And it's easy to use, low in cost, and essentially available everywhere. The specimen is loaded into the device. And the specimen can be blood, the specimen can be fine needle aspirate from a tumor, it can be a cytology smear. It's loaded into the chamber, and only 20 microliters are needed. Then over the specimen is placed a consumable, single-use consumable that has the relevant reagents for the assay dried onto it. The lid is closed, and the assay is begun by a simple touch on a touch screen. After the timed temperature-controlled incubation, the machine automatically generates cellular monolayers, captures microscopic images, including antibody-decorated cells, evaluates the images to count the cells in proportion to the binding of labeled antibodies, and from this computes a numerical result. And from that numerical result, it also is able to generate, through its artificial intelligence, an estimate of the relative proportions of the particular cell types of interest on the basis of their immunocytochemical signature, and in the case of tumor analysis, identify the most likely nature of the tumor under investigation.